Hello, everyone. Thank you all so much for joining today. We're going to be starting the webinar promptly at noon Eastern Time, so in 5 minutes. Hello, everyone. Thank you all so much for joining. We're going to begin the seminar promptly at noon Eastern Time. All right, hello everybody. Thank you all so much for joining today. We're going to get started with today's seminar. So welcome to the NOAA Omics Seminar Series. I am your host, Catherine Egan. I am the NOAA OAR Omics Coordinator, and I sit in NOAA Ocean Exploration. I also sit on the NOAA Omics Working Group as a subcommittee co-lead. So omics, which is a suite of tools used to analyze DNA, RNA, proteins, and small molecules, has become a large priority here at NOAA in the last few years. We established this seminar series in an effort to increase transparency and collaboration and highlight the incredible omics-related research currently underway both within and outside of NOAA. The seminars from this series normally take place on the third Wednesday of every month at noon Eastern Time, but we may have some seminars that are specially scheduled outside of this time frame. The seminars from this series are all made available on YouTube and posted up on our Omics website. I will drop a link to that in the chat now if you would like to view the presentations from this series. So as participants, you all are muted, but feel free to type questions into the chat box throughout the presentation. At the end, I will read the questions for the presenter to answer. Okay, so with that, our presentation today is titled Measuring the Attenuation of Marine eDNA in Nearshore Alaska by Dr. Diana Baetscher. So Diana, I'm going to give you permission to share your screen while I present you to our audience. All right, we can see your screen fine, Diana. So, okay, Diana started working with the Alaska Fisheries Science Center in April of 2021. She received her PhD in ocean sciences from the University of California, Santa Cruz in 2019, and then did 2 postdocs, one at Cornell University and another with the Monterey Bay Aquarium Research Institute. Her projects applied genetic methods across a range of ecological questions, including population genetic structure of fishes and seabirds, larval dispersal, genetic species identification, and more recently, metabarcoding of complex tissue mixtures and marine community surveys using environmental DNA, or eDNA. Diana heads up most of the eDNA research at Alaska Fisheries Science Center in Juneau, and is currently recruiting a postdoc to study changing fish distributions in the Arctic with eDNA. I'm actually going to drop a link to that postdoc position here in the chat, so feel free to apply or share widely with your networks. So with that, thank you, Diana, for presenting today. I will now pass it off to you.
Thanks so much for that intro, Catherine. Can you hear me?
Yep, we can hear you great.
Okay, perfect. I'm excited to present this set of projects, or primarily one project, from the environmental DNA program that's part of the Auk Bay Laboratories of the Alaska Fisheries Science Center. And my co-authors on this project are all folks up at NOAA here in Juneau with myself. So just very, very briefly, I want to introduce you to environmental DNA. I'm sure most of you have probably heard about it or are very, very familiar with it. Environmental DNA, or eDNA, is just very simply the DNA in cells that are shed into the water, soil, and air, such that when the organisms leave, those molecules are left behind and are able to be collected and filtered and amplified and sequenced by researchers like myself. And concurrent with the rise of all of these projects in which we collect environmental DNA is kind of a concurrent interest in what's being called the ecology of eDNA. And this sort of captures the idea that there are all these physical characteristics that affect species detection using environmental DNA, using this technique. And so in order to properly interpret our results, when we go out and we collect a sample of water and we amplify the DNA that's left in that sample, we need to understand kind of all of these physical parameters that influence that molecule. And so there are all these papers that have come out in the past, 5 or 10 years looking at habitats and temporal variation, spatial structure, tides, water stratification. So just a whole host of different physical characteristics and their influence on eDNA. And as you can imagine, that this is sort of a piecemeal project, and that these are, site-specific studies or modeling studies, but there's really been this interest in trying to figure out what are some themes that we can draw from this literature in order not to have to test all of these parameters every time we want to take an eDNA sample. And so some of those areas of consensus that have kind of emerged are that, habitat is really important, as one might also get from doing like a visual survey. You can see often communities changing across different habitat types. Depth appears to be quite important in the marine environment for eDNA and how that structures communities. Also, time of day. So there are a number of papers coming out looking at diel vertical migration and the ability to detect sort of that migration through the water column using eDNA. So, really a rich area of research right now. And the lab group that I joined last April, so basically a year ago, did sort of a project in this vein. So looking at how habitat types in nearshore Alaska influence or structure the fish community that is captured by environmental DNA. And I'm very briefly going to go through this project. Most of this work was completed before my arrival, but I think it's a great introduction to sort of what came next. So what they did was they had 9 sites of 3 different habitat types. They had 3 sand, 3 rock, and 3 eelgrass sites. And each site was sampled at high and low tide on the same day, and these were surface samples that were collected. So you have sort of a habitat and a tide variable that are the focal elements, sort of physical characteristics here of interest. And what they found, we can see in this RDA plot, is that rock habitat sort of separated out from the sand and eelgrass habitats. And so the rock habitat is in green, and the circles are the high tide samples, and the triangles are the low tide samples. So for both low and high tide, you see sort of the rock drawing toward the right side of your screen. And what was sort of interesting to me about this result was it wasn't as though there was an entirely different fish community in rock habitats, but rather that rock habitats lacked some of the species found more commonly in sand and in eelgrass. For example, like this flatfish, which is often associated with a sandy bottom. So, so it was nice as sort of a verification that given kind of the high tidal swings in coastal Alaska and the other physical characteristics sort of that might be affecting the environmental DNA, that we could have almost this gut check on our ability to detect species in sand habitats that we know to live there that are less common, obviously, in rock habitats. And that's actually been accepted at the journal Environmental DNA and should be coming out very, very shortly. Okay, so I'm going to pivot a little bit because concurrent with the interest in what I've been calling the ecology of eDNA is the methodology in the lab. And we know that there are choices that we make when we go out into the field and we sample, and that affects what species we're detecting. But there are also choices in specifically the eDNA assay choice or the primer set that we use that affect species detection. And I think about this to some extent on a continuum from more specific primer sets or assays to less specific primer sets. And the 3 that I've outlined here are quantitative PCR or qPCR, which is generally thought of as the most specific because you're targeting a single species and you're able to get, essentially an estimate of DNA copy number from that, to maybe species-specific primers. So these aren't necessarily designed for quantification, but they should give you, really good taxonomic resolution to species, for whatever your target taxa are. And then finally, maybe a more general or universal primer set, like there's just a whole host of fish primers with varying degrees of resolution across, the enormous diversity of fishes. So that's kind of the more specific to the least specific set of assays. And and we would intuitively think that the most specific assays would have the best detection ability, but that hasn't been experimentally verified actually that often, because it's just sort of a widely held assumption. Okay, so I've introduced sort of these physical characteristics that affect eDNA detection and the assay choice, kind of our lab methodology. And the question that we are investigating in this study, in which we're looking at both of these components, is over what distance can we detect eDNA from a point source? And the specific physical characteristics that I'm interested in here are tides. So Alaska has very large tides compared to other places that I've lived in the United States. And so, we think about these molecules being very, very small and subject to all sorts of water movement. And so we hadn't found much of an effect if we go back to that RDA plot from the previous study of tides, right? The habitat was really what structured the samples in that plot, but it seems like tide should have an effect on some level, and so we're continuing to explore whether there's an element of water movement that we can detect at different tides. And then we're also interested in stratification. So I didn't talk about this when I was introducing the prior study, but one hypothesis that we didn't test in that study is the extent to which water stratification, specifically in the rock habitat, where often the depth gradient was much steeper than in the sand and the eelgrass habitats, but whether that stratification actually affected the ability to collect samples that included species that are part of kind of the benthic community. So this was an opportunity to kind of look at results from that study and then have that inform the study design and what our interest was in stratification for this second study. And then finally, the eDNA assay choice, thinking about those 3 levels of specificity that I had mentioned previously. So the qPCR, the taxon-specific primers, and a universal fish barcode, with kind of the hypothesis that specificity would improve detection. The sampling location where we tested this sort of point source question is Amalga Harbor, and to orient you to where we are in Juneau, Alaska, and where Amalga Harbor is, it's here in the Alaskan Panhandle in the southeast, and it's in the inside waters. So we are not on the Pacific coast in the way that you might think about the coast. These are inside waters, but still lots of water movement, big tidal swings. And we used a setup that I'm calling a natural experiment, and it was opportunistic, but Amalga Harbor is here in this photograph, the marine environment, and Peterson Creek is a freshwater input to Amalga Harbor. And then there's this really obvious structure that are hatchery pens, which include 46 million juvenile chum salmon. And so those are placed there by the hatchery in the springtime, and there are no naturally returning chum salmon that are present in Amalga Harbor at the same time. So there was kind of this perfect window when we had this hatchery point source when we weren't going to have kind of ambient chum salmon that would change our signal. So that was, we sampled last May, and that was sort of the perfect opportunity to do this particular experiment. And so what we did was, starting from those hatchery pens, we sampled along a 2-kilometer transect, and we sampled every 80 meters, as you can see in this plot. And the depth changes quite substantially, especially as you leave Amalga Harbor. So we were interested in this depth gradient, and we took CTD casts at each of these sampling locations as well. We sampled at both incoming and outgoing tides, so getting at that tidal question. Here in this plot, you can see where in the tidal cycle we sampled. So the incoming tide was in the morning, and the outgoing tide was in the afternoon. And here's that large tidal swing that I was mentioning. So on May 10th of last year, it was a negative 0.8 foot low and a 14.8 foot high. We collected these samples by boat from the surface, and you can see them tucked here in their Whirlpack bags in this cooler. There were 3 1-liter field samples collected per location, per tide, and they were collected consecutively. So, back to back, 1, 2, 3. The photo really nicely shows the turbidity gradient between the onshore and the offshore samples. So the onshore samples or nearshore samples are the turbid ones at the top. And then you can see it getting progressively clearer and clearer water as we got offshore. And for anyone who's filtered water, the nearshore samples were very slow. Despite being not actually terribly silty, the filtering went very slowly on those. Okay, so going through the lab methods here, we had, from this liter of water, we filtered it down and then preserved the filter in Longmire's buffer, which is great and stable at room temp. Then we extracted the DNA from that buffer, and then from that extraction, we were able to perform qPCR, the salmonid specific metabarcoding, so that's our taxon specific metabarcoding primer, and then 12S metabarcoding, which is our universal fish primer. We did 3 replicates, as I've indicated, of each of those assays. And so when I'm showing plots in the results that have a mean, that's taking the mean of 9 total replicates per location per tide. For folks that aren't terribly familiar with metabarcoding, I'm just going to give a super brief overview. So again, that same DNA extraction, what we do in metabarcoding is use the primers that we've selected. So for example, our universal fish barcode to target a particular DNA sequence, and then we're able to amplify that, sequence it on our MiSeq, and then essentially sort the sequences by similarity and assign them based on an identity threshold to a reference database. So you can just see that the sequences are grouped by similarity here, and then we can put a fish to that sequence using all of these publicly available reference databases. All right, so I'm going to get into the results, but I just briefly want to remind us that we're looking at the tides, and we're looking at whether detection distance changes using each of our different eDNA assays. And then we're looking at stratification. So is surface sampling affected by the mixing of the surface and the lower layers? And then finally, does specificity of our eDNA assays improve detection? And I'm starting here with the most specific of our assays. So this is the qPCR. On this plot, the x-axis is the distance from the hatchery pens from 0 to 2,000 meters. And that's actually very consistent with the rest of my plots. And then the y-axis here, it says DNA copy number, and that's log scaled, and that's the mean for each of those 9 sets of replicates. And what you can kind of see is there's not much difference between the incoming tide and the outgoing tide. So incoming tide, the red circles, and outgoing tide, the blue triangles. And if we fit a linear model to those log scaled points, we see that the 95% confidence intervals are not entirely overlapping, but very much overlapping for the majority of this, and that there's no obvious difference between the tides here. We also see that it's kind of a tighter fit toward the top to about 1,000 meters, and then it gets a little bit noisier past 1,000 meters. And so one of the things that we were interested in is whether the bathymetry of this transect would be affecting our surface qPCR signal. And the only thing that strikes me is kind of this shift from that the data points being relatively tight around the line to kind of being noisier after about 1,000 meters, and whether that corresponds to some of the drop off in depth, or perhaps more likely essentially leaving the shelter of Amalga Harbor and being more in the channel, so potentially more subject to water movement. But that's something that we're interested in exploring potentially for further with some transport modeling or hydrodynamic modeling. But there's not a great surface qPCR to bathymetry signal, and certainly not one at 500 meters depth where you kind of see this first drop off in the bathymetry. Okay, I'm going to hop into the salmonid specific metabarcoding results. So this shows the distance from hatchery pens on the x-axis, and then the mean number of sequencing reads across replicates on the y-axis. And the top plot is the incoming tide, which was in the morning, and then the bottom plot is the outgoing tide in the afternoon. And the colors correspond to all of these species of salmonids, but the one that we're interested in, I've colored in kind of this dark salmon. So that's our chum salmon. And you can see a pretty clear, although somewhat noisy, pattern of more sequencing reads closer to the hatchery pens, and then it kind of diminishes as we get further and further from the hatchery pens. And that you see that both in the incoming tide and the outgoing tide, but there's certainly some variation across the tides. And I'm just going to change the plot to be a proportion of sequencing reads, because that kind of highlights something that I think is relatively important. But the idea that when you look at the proportion and you have really low read depths, it's easy to get these sort of signal spikes. So here in the purple and red and yellow, we have coho and king and sockeye salmon, which may not be very abundant, but if we're looking at the proportion of sequencing reads, they're sort of over amplified by that signal in comparison to looking at the number of sequencing reads, in which it's still relatively modest. And then if we look at our 12S universal fish barcode, we can see that the chum salmon is still in that same color, and that we still get that consistent pattern of decreasing number of chum reads with increasing distance from the hatchery pens. But we get kind of this blue set of taxa also cropping up. And here I'm going to use that proportion to sort of highlight that. So I think this shows it more clearly that we basically in the light blue, in the dark blue, and in the kind of navy blue, we have herrings, smelts, and cods. And that's kind of a more pelagic and maybe more marine signal. And we see that very clearly occurring much earlier in our transect in the outgoing tide. So right around here, where on the incoming tide, we have this very strong chum salmon reed signal up until about maybe 1,200 meters, and then it drops quite dramatically, and then we get kind of what I'm thinking of as this more marine offshore signal. And then in comparison, in the outgoing tide, we have that signal kind of cropping up at like 800 meters. So pretty different across the incoming and the outgoing tide, and actually different than what we might have expected, because on the outgoing tide, at least as we were thinking about it in our experimental design, the water movement would be sort of flowing past the chum pens and drawing maybe that eDNA further into the channel. So we were expecting the signal to perhaps be stronger further out on the outgoing tide than on the incoming tide. And that's not what we're seeing based on this 12S universal fish metabarcode. But I'm gonna describe some of our hypotheses on that front in just a few minutes. But at first, I want to take kind of this detour and talk about the eDNA assay choice, because I think it's useful to perform essentially a side-by-side comparison of our qPCR, our salmonid metabarcode, and then our 12S metabarcode. And what I've done for comparison, so this is that same qPCR plot that I showed previously, and then I've taken just the chum salmon reads from our salmonid metabarcode and just the Oncorhynchus reads from our 12S fish metabarcode for comparison. And I think what's useful about this side-by-side comparison is that the shape of the line and the plots, although there is more noise or wiggle, certainly after 1,000 meters, and perhaps more so in the metabarcoding data, they don't look that dissimilar. And the R squareds, if I add those in, actually for the 12S metabarcoding data aren't horribly different than for the qPCR. And I wanted to just make this point because qPCR is really thought of as the gold standard, but there is a lot of additional information that can be gained from using metabarcoding, as we saw from the sort of taxon proportion plots. So, to the extent that it's feasible, thinking about the ways in which metabarcoding can be semi-quantitative or sufficiently quantitative for some of the purposes of a study like this, I think is quite useful as one of those lessons that's transferable to other projects or future projects, certainly in our lab group. So sort of quick summary, the qPCR is the best fit for this sort of log-scaled and linear model, but metabarcoding using the sequencing reads in a similar format recovers a similar trend. All right, and then I want to pivot again and talk a little bit about the physical characteristics of water stratification. So I mentioned that along that transect, we took CTD casts at each of our locations during both the incoming and the outgoing tides. So I'm going to show you plots where we're looking at temperature versus depth and then salinity versus depth for all of our transect. And this plot is arranged very similarly to the sequencing data, except here the y-axis is depth, and the deepest I've plotted is up to 10 meters. So sorry, this is the temperature plot, with the warmer temperatures being kind of the yellower, greener colors, and the cooler temperatures being the purple, blue colors. And what I want to focus in on here is the depth of the surface layer, right? Because that's where we collected our samples, just sort of leaning over from the boat. So we're really only sampling, the top meter of water. But the depth of the surface layer on the incoming tide and the outgoing tide at least in temperature, looks to be kind of mixed to about 5 meters. And on the outgoing tide, we can really clearly see that we have the warmer water, likely just from insulation on top. And then if we look at the salinity data from that CTD, we can see that on the incoming tide in the morning, there's the depth of the surface layer that's to about 2.5 meters, whereas the depth of the surface layer on the outgoing tide is to about 5 meters. So there's actually a deepening of the surface layer that occurs on the outgoing tide, or perhaps more likely just because it's in the afternoon. And I was thinking about how that might be influencing the results that we're seeing with our metabarcoding data. So here's sort of my hypothesis about integrating the CTD data, so the water stratification, with what we're seeing in our metabarcoding results. And I'd be very curious if folks have a reaction to this or think that this is something that they've seen in their own data. So basically, we're sampling from the surface layer, and it's well mixed to about 3 meters in the morning on the incoming tide. And then in the afternoon, we have a warming of the surface water temperature, and we have increased stratification along with that warming. But we also have a deepening of the mixed layer, as we're seeing in those salinity plots. And that's likely, at least in my experience, caused by wind, right? It's not caused by a temperature element. It's, most often that the mixed layer is deepened by wind. And so it seems possible that the deeper surface layer in the afternoon on the outgoing tide may actually dilute the chum signal when we're further away from those hatchery pens. And so this is what I'm talking about here, where on the incoming tide in the morning, we see the strong chum signal, and then it kind of drops off at 1,200 meters, sort of where the signal got noisy in the qPCR. Whereas in contrast, on the outgoing tide, we have that marine, that herring smelt signal crop up much sooner. And my thought is that it's possible because we've deepened the surface layer, and there's more volume of water that's sort of mixing with that chum DNA in the afternoon. Our amount of chum DNA in that water sample, in that same 1 liter, is just diminished because it's been diluted. So not a perfect explanation necessarily, but it seems plausible that you have an element of a changing of the mixed layer depth that's actually influencing this result, especially because these samples are collected in the surface. Okay, so if I want to summarize how physical characteristics and eDNA assay choice affect detection in this particular study, for tides, we did not see a difference for qPCR or the salmonid metabarcoding, but we did for the 12S fish metabarcoding, and that's seemingly likely because with the more general metabarcoding primer set, we're actually able to pick up the kind of background fish community that is being masked by this really, really large point source of these chum salmon. So, so we're thinking of this as sort of like a signal to noise ratio problem. In terms of stratification, is our surface sampling affected by mixing of surface and lower layers? It seems like actually it could be, and it could be affected by this deepening of the surface layer in the afternoon. But that's something that we're really curious in going back out to test further, and hopefully we'll have an opportunity to do that again in May using those same hatchery pens. And then finally, the question of eDNA assay choice for qPCR versus salmonid specific primers versus sort of the universal fish metabarcoding approach. Does specificity improve detection? So not substantially, if at all, in our study, and that seems to be the case in part because we were detecting chum salmon right up to the end of our 2-kilometer transect. But it's possible, had we gone beyond 2 kilometers, the specificity benefit of something like qPCR could have cropped up. So, I think it's, does it matter for a particular distance is going to depend on the strength of the point source and the length of the transect. Okay, and then trying to integrate some of these ideas into recommendations or future directions for these questions of ecology of eDNA and lab methodology. So the first point is that eDNA detection is strongest closest to its source. So that's something that's very widely accepted, and I think that actually is one of the great strengths of using eDNA as a sampling technique, is that you're not getting as many sort of far away signals as you might think. But our detection from this very large point source did extend 2 kilometers. And so although, right next to the chum pens, we had this really, really strong chum salmon signal, we were picking up that chum salmon far away. So, so I think, there's a lot more to be done with questions about biomass and transport, but we do have this dynamic of when you have a really strong signal, it's likely that you're close. Whoops. Okay, and then the second point, the signal to noise ratio changes with primer specificity. So this is the point I was just making about using the 12S fish metabarcode and being able to see kind of the background herring smelt gatted signal when the chum salmon signal dropped off. So if you're using a more specific primer, for example, qPCR, then you're not likely to see that, right? Because you're only able to see the chum salmon. So what this could be actually used, or this information could be leveraged to think about, if you're using a specific primer, then maybe something like time of day or tide may not affect your results as much, right? If what's affecting your results is kind of the displacement of one community with another, then you're only going to see that if you're using a less specific primer set. And then finally, the third point I wanted to make here is that metabarcoding read counts in our context do appear to be semi-quantitative. And that because there's all this additional information that you can get from metabarcoding data, to the extent that we're able to use some of those metabarcoding tools in lieu of qPCR, I think it really is a rich area for continuing to develop our understanding of that relationship between the read counts and the quantitative nature of metabarcoding. All right, for next steps, what I alluded to previously is thinking about kind of the water movement in and around this area and how that might be affecting our eDNA transport, particularly around 1,000 meters, or as we start to leave Amalga Harbor, whether there's cross sort of transport that could be changing, the direction of eDNA flow. And then more broadly, kind of what are the transferable lessons to other marine habitats, including things like stratification, particularly about surface sampling and when the mixed layer is deeper or shallower, and then the metabarcoding versus qPCR sensitivity. So this question of what's your goal? Is your goal a single species goal, or is your goal more of a community goal? And can you get all of the information you want or need from metabarcoding? And this is actually just one piece of sort of the eDNA work that we do at the Alaska Fisheries Science Center. So I wanted to just very briefly highlight the other projects that we're working on. This is, clearly part of our method development and understanding the ecology of eDNA. But we have this postdoc position that Catherine mentioned, where we're looking for someone to come on and help us think about how to assess rapidly changing Arctic communities using eDNA. That's a collaboration with folks at PMEL in Seattle. And then we're really interested in piloting some studies on the bottom trawl survey. So figuring out, how eDNA can be kind of a complement to pulling nets, and then potentially leverage that to extend survey range. And then finally, in locations where we really can't pull nets because of rocky substrate or other habitat characteristics, can we use eDNA perhaps in conjunction with other techniques, but to characterize the species composition in some of those untrawlable habitats. And we're doing this work in partnership with folks from USGS and the Northwest Fisheries Science Center and PMEL I mentioned, and then we have some students that we've worked with at both UAF and UAS up here in Alaska that are some fantastic partners in thinking about these eDNA questions. With that, I want to thank my co-authors on this and the NOAA Alaska Regional Office for funding. And I'm happy to take questions in the chat or feel free to shoot me an email. Thank you so much for your time.
Excellent. Thank you so much, Diana. A fantastic presentation. We're going to go ahead and jump into the questions because we have a lot of them coming in. So Diana, I'll read them out loud, but you should be able to see them here in the questions box. So our first question here is from Catherine. What pipeline did you use for the 12S metabarcoding? Did you adjust parameters to try and improve detection?
I used sort of a series. So I used cut adapt to remove primers and then data to identify sequencing variants, and then basically use the output from data 2 as my blast input. So in terms of adjusting parameters to improve detection, I guess the only thing that I adjusted of note was I used like the pseudo pool option on data 2. I'm not sure how deep into the weeds folks who do this work know that there are a lot of weeds. So yeah, so I basically, I didn't do anything strange, I don't think, in terms of normal metabarcoding pipeline in order to, improve detection. This is a pretty standard pipeline that we've used in this lab group and I've used previously, and I think is pretty well established in the literature. So I think the detection wasn't high graded in any way. Sorry if that wasn't exactly what you were looking for.
Excellent, thanks, Diana. Okay, we have a multi-part question here from Krista, so I'm just gonna break this one up. And so she says, really nice talk and study, Diana. For the chum net pen and communities surrounding them and outward study, if one is interested in the communities around these net pens, is the chum signal overwhelming what you can actually glean from the nearest communities using eDNA metabarcoding? In other words, are sequencing reads capturing as efficiently critters that are much less abundant than chum? And then after that question, we'll go into the second part of that question.
I think so this is definitely thinking about the 12S metabarcoding data. And I think, right up against the chum pens, you are having this masking effect, right? So that's where you see nearly all of the sequencing reads going to chum. I did have this thought that I haven't exercised yet, but of essentially subtracting out all of those chum reads and then doing like an RDA or an NMDS of the community remaining in the reads, because you can sort of see it, but it is totally masked by the chum that you have, perhaps a different onshore versus offshore species composition. So, some more amidites and things onshore, and maybe some more rockfishes and other things offshore. But with the sequencing reads, there's always the trade-off. So if you have a big signal that's absorbing a lot of your sequencing, then you're not seeing necessarily everything else around it. And I think that's what you get basically when you move further out in the transect. but the extent to which we're seeing the community in sort of its fullest form right next to the chum pens, I think that's definitely more questionable just because of that signal masking.
Awesome, thank you. And I think you touched on it a little bit, but the second part of Krista's question is, lost it there. Okay. What molecular approach would you take to effectively remove chum from the data or amplification to begin with, to ask the question of what communities look like nearest and furthest from the net pens?
Yeah, I mean, that's a great point, because what I was suggesting of basically subtracting the sequencing reads, won't solve the problem of you've already done the sequencing and are missing things, and we know that. And so instead, you could use some sort of blocking primer, although I've heard those are sometimes not terribly effective. But yeah, you would want to use something sort of upstream of your data analysis and your sequencing, so that you could actually remove the chum if that wasn't what you were interested in. I think the flip is that you could probably sample Amalga harboret almost any other time of year, and then you wouldn't have that overwhelming signal. So we've clearly sort of artificially changed when we've sampled in order to do this transect. But in most cases and in most papers, what they're looking at in terms of horizontal transport is very much habitat associated. And so there, I think what you're seeing across habitat types is a very clear signal that you're not sampling something really far away. You're sampling the community that's associated with that particular habitat. And those studies show that you can use eDNA to do that on very small spatial scales.
Great, thank you so much. Okay, moving on to our next question. This one is from Linda. For the comparison of the different detection methods, could the lack of significant difference change when trying to detect a less abundant individual? So you had millions of chum to contribute to eDNA material, whereas if you were trying to detect a rare species, qPCR might be more sensitive.
I absolutely agree with that. Yeah, so our point source, 46 million chum salmon in a pen, that's just a huge input. And for rare, I think for rare detections, qPCR is absolutely, the gold standard, and that, your metabarcoding approach probably wouldn't be the right tool for that question.
Thanks, Diana. Okay, a few more questions here. We'll have another question from Catherine. Do you have any data or studies planned to look at the impact of biomass, e.g. number of fish, on detection?
Great question. So yeah, this is something that I've been thinking a lot about, because what you want to be able to do is disentangle the amount of biomass and the distance that you are from that source, right? If you have a huge amount of biomass and you're further away, then it could look the same as if you have a smaller amount of biomass and you're not as far away. And so to the extent that we're able to do that experimentally, I don't know that we will necessarily use the same setup, but we have, as part of actually this sort of postdoc funded project, some aquarium experiments planned. And our goal there is to do much more of that fine tuning. The challenge is always, okay, how then do you transfer that to the actual, the middle of the Bering Sea or something? But I think that's a great question. And that some of the folks that we're hopefully working with on that aquarium study are interested in things like, does the shedding rate change with increases in temperature? And so can you actually start to control some of the additional variables? So it's not just biomass, but it's, shedding from that biomass, right? So that's a really great question, and I think a really important line of inquiry, especially for us at the Alaska Center, to be able to make this a really useful tool for some of the other folks that we work with, especially on some of the larger surveys.
All right, thank you. Okay, on to our next question from another Diana. How would you apply what you've learned from these comparisons to attempts to detect rare species, such as ESA-listed salmon and steelhead with very low abundance, in nearshore or estuarine environments? For example, sample at incoming tides, or when mixing is best and surface layer is shallowest. Other strategies?
I mean, I think one of the previous questions alluded to this. If you don't have a large point source, or if you're not interested in broader community and what you're interested in are, ESA listed Chinook salmon, then it seems as though qPCR, would probably be your preferred method. The extent to which, sampling across some of these different tides or times of day would make more or less difference, hopefully would be mitigated by using that super sensitive genetic assay. But we always are challenged by the rare things. And so if you're looking for the single fish, then that's going to be harder to find. Or it could be a much more complicated question if you have a mixed stock and you're looking for the handful of ESA listed fish among a bunch of non-ESA listed fish. That's sort of a whole different realm of using eDNA in combination with population genomic techniques. And I think that's also a really rich area of development in eDNA, but not something that we're currently looking at in the context of this study or our work. Yeah, great question though.
Awesome, thanks, Diana. Okay, a few more questions here. Our next one is from Linda. Really interesting work, thank you. In the first study, how far apart were the different habitats, sand versus eelgrass versus rocky?
Oh, good question. This is going to test my Juneau, my local knowledge. Let's see. I mean, they basically spanned, I think, probably maybe 20 miles. So here in Juneau, our road system is pretty limited, and basically it went from all the way out the road to sort of near the lab. So I think, yeah, probably around 20 miles, but they were scattered throughout such that some were closer together. So there were little clusters. Yeah, they looked at, I think they looked at distance, but really what was structuring it was because you actually had sites that were quite far apart and quite close together for each of the different habitat types. What was really structuring it was the habitat itself and not simply the distance across habitats. And this is something I literally was just looking in the literature last night because I'm really interested in the extent to which prior studies that have looked at horizontal transport have actually sort of been confounded by this habitat variable. So, if you're sampling and you're trying to say that there's a limited movement of eDNA, from one side of the bay to the other side of the bay, but you have a reef on one side and eelgrass on the other side, what you're actually sampling are two different habitats. So I think one of the things that we're constantly grappling with, and you saw this with sort of the tide effect versus the surface layer deepening effect, but how do you disentangle variables when you're performing essentially natural experiments? And what I think we're doing better as an eDNA community is identifying when those things are sort of confounding, and then hopefully designing the next study to more intentionally test, one thing versus the other. So hopefully that's what we did here.
Thanks, Diana. All right, I think you touched on this a little bit from some past questions, so I'll read it, and if there's any additional information you want to add to this, how do you think these methods would apply to a weaker or non-point source eDNA signal? And this one comes from Libby.
Yeah, and I think there has been a lot of interest in this. So I think that these results are not going to be perfectly transferable, especially kind of the metabarcoding approach or potentially the sequencing reads. I've seen rarer things in metabarcoding data, of course, but it's harder to be able to quantify, kind of like, are you, like I've mentioned, are you close to that? Is it lower biomass? Are you far from it? So, it's just much more difficult to sort of bound what you're seeing if it's a less significant point source. Whereas if you use qPCR, because you know what you're interested in, and that's something that may not be terribly abundant, then you're able to say something pretty definitive about the DNA copy numbers. It doesn't solve the problem of, is it a single individual? What was the temperature? What was the shedding rate? How far were you from where it was sampled? But if what you're interested in is rare detections or less abundant detections, then qPCR can be a way to go. If what you're interested in is more general community, and that you're not necessarily going to have an overwhelming point source, but it's sort of this composition question, then metabarcoding is sort of a more obvious tool in the toolbox, I would say.
Thanks, Diana. So folks, we only have a few minutes left, and there's a lot of questions coming in. So if you haven't done so yet, jot down Diana's email so you can reach out to her. So we'll just keep going through the questions until we reach 1 p.m. Eastern time. So our next question is from Kristen. What are the prospects of using copy number in qPCR or read count in metabarcoding for abundance?
I mean, I think there's been some use of, I don't know, it's sort of a squishy abundance that people have used. It's saying, relative abundance and not necessarily putting a specific bound on it, or it's, a study that uses relative abundance and has tested that in some capacity, so they know what it means. I mean, I think I've seen this more often in metabarcoding, like diet studies, where you control, relative proportions or something of that sort. Yeah, to use it for straight abundance, I think you run into a lot of the problems that I've mentioned before of, if you collect a liter of water and you get a number of DNA copies with high precision using qPCR, you still don't know, how many organisms that corresponds to. And what we're hoping to do, actually, as part of this postdoc position in thinking about kind of the changing Arctic communities, is to put better bounds on that using this aquarium study. So, as I mentioned, shedding rates and temperature dependence and biomass is one of those elements. But you also have the problem of, it can be biomass, but it could be many small fish, or it could be fewer large fish. And for our purposes at the Alaska Fisheries Science Center, that distinction actually matters. So, yeah, I don't know that I've seen anything where I'm convinced that there's like a perfectly generalizable way of doing that yet. But I think a lot of us are really interested in that, and that's a little bit of the holy grail of eDNA is being able to make that jump to abundance. Some folks are trying to do it with kind of a population genetics type approach, where if you can figure out the number of haplotypes and the number of individuals, then you can back out, okay, I have, whatever, 20 fish. But that's a very involved and very different approach. So maybe not a satisfying answer, but we're working on it, I think, is the short answer.
Excellent. Thank you so much, Diana. Folks, I'm so sorry we were not able to get to all of the questions today, but like I said earlier, please reach out to Diana directly for more information on her research. So Diana, thank you so much again for presenting today, and thank you everybody for joining us today. I'm going to give you all just like my little ending spiel here. So the NOAA Omics Seminar Series, as I stated earlier, takes place on the third Wednesday of every month at 12 p.m. Eastern Time. We actually have a specially scheduled seminar taking place next week on March 24th. It's titled Deep, Dark, and Diverse, an exploration of hydrothermal vent plume community composition and function. Dr. Matthew Hark from the Gloucester Marine Genomics Institute will be our speaker. You can register for that seminar up on the NOAA Omics website. Additionally, there will be no seminar for April, but we will resume again in May. So Diana, thank you again, and thank you everybody for attending today.
Thanks so much, Catherine.